Hello, and welcome to this third season of This is Epigenetics, the bilingual scientific podcast presented by the Canadian Epigenetics, Environment and Health Research Consortium, or CEE-HRC podcast, hosted by their trainees. We interview renowned Canadian scientists to talk about their career, their research, and of course their passion for epigenetics. We hope you enjoy this episode. Hello and welcome. I'm Sabrina, a PhD candidate in the Couture Lab at the University of Ottawa, and I'll be your host for today's podcast. I'm also joined by Laura, a postdoctoral fellow at the Dana-Farber Cancer Institute, who will be co-hosting this episode with me. It is an honor to have with us today Dr. Paola Marignani, a full professor in the Faculty of Medicine at Dalhousie University. Dr. Marignani holds a primary appointment in the Department of Biochemistry and Molecular Biology. She completed her postdoctoral training at the London Feldtanenbaum Research Institute, the Ontario Cancer Institute, and Harvard Medical School in the Division of Signal Transduction. In addition to her academic accomplishments, Dr. Marignani is the founder of Canadian Women in STEM, a national initiative focused on advancing equity, diversity, and inclusion across Canada's STEM landscape. This network includes over 2,000 self-identified scientists across the STEM spectrum and has been instrumental in supporting the National Sciences and Engineering Research Council of Canada's Dimensions EDI program. Dr. Marignani also contributes her expertise beyond academia. Since 2021, she has served as a mentor scientist and business strategist with the Creative Destruction Lab, supporting innovation and growth in both the prime and cancer streams. Thank you so much, Dr. Paola Marignani, for joining us. We're truly excited to have the chance to speak with you today. To start off, could you tell us what sparked your interest in science and what motivated you to pursue a career as a researcher in epigenetics specifically?
Well, my interest in science started when I was really very young. I was probably 5 or 6 years old. And I was that kid who turned over rocks and was hunting for all sorts of things in nature and trying to understand what this thing was. And, I was the one who had tadpoles and tried to chart the growth of a tadpole to a frog without actually knowing what it all meant. And I was that kid who had all sorts of crazy things in my backpack that I got into trouble for quite a bit when I was younger. But I've always had this curiosity. And so I didn't really know what a scientist was. All I knew was that I really enjoyed nature and trying to figure out nature. And so it really wasn't until I was 8 or 9 years old and my family, we went back to Italy for vacation and we went to Florence. And at that point, my parents introduced me to the arts and, of course, Leonardo da Vinci. And that was really when I realized there's people who do this stuff for a living. And it was the first time I ever heard the word scientist, because Da Vinci was being referred to as a scientist in a lot of the galleries that we were walking through. And I thought, this is inventing and discovering and creating, and it was art and this other stuff that I really love to do. And that's the first time I actually heard the word scientist, was at that moment. And not knowing exactly what the scope of that was, I actually proclaimed at 8 years old that I was going to become a scientist. And my family didn't even know what that was, right? Because we're immigrants. So they didn't even know what that was. They just thought this little girl wants to be a scientist. She's going to be a painter. She's going to be a sculptor. She's going to be, someone who does this sort of stuff. And so as I'm getting older, I'm understanding what more science is about. I then started my path towards becoming a scientist. And there was no precedence. There was no one in my family who had taken this path before. And because, again, we were immigrants, we're the first generation to go to school as well. So there was no template for me to follow. And when I was young, there wasn't even a template or a person within the Italian community where I grew up that knew how to take this path. So it was always a challenge, but for me, it's always about the excitement of discovery. And I had that as a child. And I always say to myself, the day that I lose that childlike approach to research, where I'm super excited to get into the lab in the morning and super excited every time I see data, even though I have no idea what it means, or even if the experiment fails, it's already a path forward, right? The day I kind of lose that child approach or vision of discovery is probably the day that I walk away from science, because I no longer have that joy at looking at the research or joy at looking at nature.
So it's truly that curiosity and passion
Curiosity.
that motivated you to pursue a career as a researcher.
Yes, not knowing what it was, because there was no one in front of me to say, this is how you become a scientist, this is what science is, right? So it 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 truly was a very, very curvy path. There's no straight lines on how I got here. There was nothing direct about it. So it's a passion, right? It's a joy and a passion.
Yeah, I completely relate. So related to your path to becoming a researcher, could you expand or walk us through your professional journey so far?
It started off as, like everyone, an undergraduate degree in either biological science, but I did my undergrad at the University of Windsor. So there was the biology program, and then there was the ecology, which is now environmental sciences. So I started off in your basic biology, environmental science with chemistry. And then I did my master's at Western, at the University of Western Ontario, in neurobiology. So I actually have a pretty deep knowledge of brain and brain structure and function. And then I moved to McMaster University to do my PhD in membrane biophysics. So with that, I changed disciplines every step. So for every degree, I actually changed directions. And then I did my first postdoc at Harvard in the Division of Signal Transduction. And that was really my first exposure to molecular biology and to signal transduction. I spent about 4 years there, and I came back to Canada, and I went to the Samuel Linenfeld Research Institute to do a second postdoc to learn mass spectrometry. So this was the height of mass spectrometry and proteomics. It was just at the forefront of the proteomics. And I developed a gel-free method of mass spectrometry. And then I did a third postdoc at the Ontario Cancer Institute, and that was for 1 year, where I actually took all this, my undergrad, my master's, my PhD, my first postdoc, my second postdoc. I took all those technologies and those tools and started thinking about the context of signal transduction in cancers. It was that switch over to Ontario Cancer Institute where I started thinking about all of the signaling in cancer. And then I got my faculty position at Dalhousie University, where discovery I had made as a postdoc at Harvard, I brought it back to Canada with me, and I had been working all along, but now I put it in the realm of cancer, and my entire lab was built on this tumor suppressor kinase called LKB1. And so that was in 2003. And so my journey is not straight. I didn't do an undergrad, master's, PhD in the same discipline. I actually changed disciplines along the way. And in part, it was because I would I did my master's, and then I had more questions. It's like, well, what is this black box? How do I figure out what it is? And that led me to the membrane, where a lot of receptors are, we know receptors are in the membrane, right? So it led me to the membrane, and it led me to lipids. And then I thought, okay, I've got a handle on the lipids, I've got a handle on the membrane, but what happens? What happens when a ligand binds to a receptor? And this was the early days of signal transduction. So the next step, what's going on right after a ligand binds to a receptor? And that's when I went to Luke Cantley's lab at Harvard, where, he was the person, the PI3 kinase, AKT, PDK1. It was really at the start of all of the signaling world, and all the domains were being identified. Tony Pawson in the SH3 domains and the SH2 domains, Tony Hunter, all of these people were discovering all these different domains, right? And so going there, I basically jumped into this fire. Because remember, I'm not a molecular biologist. And suddenly I'm surrounded by molecular biology. So it was a very steep learning curve to go from lipids and biochemistry to molecular biology. And I just embraced it. Like I jumped off that cliff and I just dove right into the world of molecular biology. And there I developed an in vitro expression cloning protocol that eventually was purchased by Promega. But I ended up, building some technologies and tools that even to this day in my lab, part of my mandate is to be able to answer our big science questions. Technologies facilitate that. So for me, technology is not biology, but technology helps me and my team get to the biology. So I'm really, I try and train my team to be nimble and quick when new technologies come on. Let's try and get them in. Let's try and adopt them, adapt them for our needs, and we move on, right? So the journey has not been straight. And I think my default setting is not to take the straight road. I think my default setting is to take curves and to I don't want to say meander, but if I have a question and what I'm doing today doesn't answer it, I will turn and take on a technology to try and answer it. I'm not worried that it may fail, because it may fail, but it may succeed. So I'm more along the lines that might actually succeed, right? A lot of people think things, if I take that chance, it's going to fail. I'm like, okay, at least you took the chance, right? So it's not a straight path.
I find it really interesting how you took bits and pieces of knowledge that you acquired at different steps of your journey to build your specific research interest that you're still interested in your lab now. So I find that.
Yeah, and there's one technology I did in my master's. I was looking at dopaminergic receptors in the brain, and it's a specific type of radioactive assay. And fast forward, we're doing kinase assays in the lab, and I'm thinking there's got to be a better way to do this, and a more efficient way, a safer way. And so I thought, when I was doing my receptor binding assays, we were using these manifolds and all these different membranes. I thought, how can I adapt this technology for radioactive work, kinase assays? And I did. And so, as I'm trying to build this little contraption and I'm going to the arts and crafts store buying things to parts and pieces, a woman asked me, so why do you need this little thing to cut circles? I'm like, I thought, do I tell her to do radioactive experiments, or do I just tell her I'm doing arts and crafts? I didn't know how to answer that question. But I think it's important as scientists that we take all the different technologies and basically, even if they're old, very old traditional technologies, we use them today and sort of integrate them into our modern, really, cutting edge technologies. So it wouldn't be unusual to be in my lab and see very old traditional pieces of equipment, because there is no modern ones. So they're very old ones from the 60s and the 70s, and very cutting edge technology. They'll be side by side, right?
I think sometimes you can also answer the question better with some of the traditional methods as well in some cases.
Yeah. And I think that's with a lot of things, but this is why with my trainees and my team, we'll sort of try and de-risk a lot of the experiments and see what is the better approach. And sometimes it is the older technologies, and because people want to always use the cooler, more modern things, sometimes I have to convince them to use the older stuff. And then when they use it, they're like, wow, this is pretty cool. Yeah, it's pretty cool.
Yeah. So I guess another question for you, Paola, is, you said that you've kind of gone from different topics. Why in particular did you choose the specific field that you're in now? Was there something about it that really grabbed your attention?
Well, cancer, I've gravitated towards the cancer because when I first started piecing together all the signaling and how LKB1 is involved in signaling, cancer was at the forefront. And at the time, cancers in general were thought to be very complex. And today we know they are. They're very complex, very heterogeneous. And because of the complexity, it's sometimes very difficult to try and understand what role your proteins or your genes are playing in cancer. We know a lot. I mean, a lot has been discovered, but I think today more than ever, because of the technologies and the granularity that we can get to when it comes to individual cells and to the transcriptome and the proteome, I think we are at the start of a new age for understanding cancers. And so I started in the cancer field when I went to the Ontario Cancer Institute, and I just fell in love. It sounds horrible, but I fell in love with the disease itself because of its complexity. So I was coming in at it a very pure, why is it happening? Why do we get cancer? Why can't we solve these cancer problems? And for me, it boiled down to trying to understand how normal cells function to then better be able to understand how cancer cells function and why cancers happen. So I was coming in really from a basic science, wearing a basic science hat. Now that I've been in the domain for a long time, it's surprising how much we know, and it's surprising at the same time how much we don't know. And it becomes even more obvious now with some of the technologies that we're using. You know, my lab does single cell RNA-seq, we actually do it in-house, and all of a sudden we're getting information that one would have thought it would have been discovered years ago with bulk sequencing, with traditional transcriptomics, and now we're discovering more things. We're even discovering new cell types that people had thought we had already discovered all the different cells in a tumor or cells in a body. And now with these new technologies, we're finding out more. So that just tells me there's just so much work to do. You know, there's just so much work to do, right? And so being passionate about this particular family of diseases, and I find there's other diseases that complement cancer or are inversions of cancer. So the signaling pathways that are involved in disease, cancer progression or tumorigenesis might be opposite in another disease. So they're downregulated or they're behaving differently. So it's like you're studying one type of family of diseases, cancer, you can get information about some of the neurodegenerative diseases. And that loops me back to my master's degree where I did quite a bit in the brain. So for me, I was drawn to cancer because of its complexity.
And do you think throughout your professional journey so far, you've had like specific role models or, you know, scientific heroes that you kind of have helped you move throughout your career?
This is a difficult question for me to answer because I don't know if I've actually had role models where I said, oh, I want to be like them. In part because as a young woman at the time when I started graduate school in the early 90s, there just wasn't anyone in front of me, and we didn't even know the roles women played in all of the discoveries that we take for granted today. We're only finding out now the roles that women played in a lot of these diseases or devices that everybody's using on a daily basis. We're only finding out in the last 6 or 7 years the role women played. And at the time when I was going through school, there wasn't role models that I could look at. And, you know, access to social media didn't exist. Hate to tell you, the internet didn't exist. Googling didn't exist. And so even to know who was out there, other than, you know, in my mind, it was Da Vinci and experiencing Da Vinci in Florence at 8 years old. That was, for me, probably the galvanizing moment in my life that drew me to science.
And did you at any point, we're all in the academic career trajectory, but did you ever at any point think about other types of careers, such as industry, or where you could still do science, but I guess in a slightly different way?
Well, in my career path that I didn't really talk about is I was already a full professor. I was already, I think 14 or 13 years into my academia career, and I went back to school to get an executive MBA from the Ivy School of Business at Western. And so that was 4 days a week for 16 months. I would work on my executive MBA, and in part it's because I believe science is actually a business. We are running businesses. Our labs, our academic labs, are in fact small businesses. In fact, in some places they're large businesses. And so I have always had sort of a business acumen, some skill in that domain. So having the executive MBA has helped me because I did have a consulting company on the side, and now I have a small biotech startup company. So I do believe business and science go hand in hand. And in fact, even one of my graduate students, after they completed their PhD, they went off and they started a biotech company and have just launched their first product. So I believe that business and science and academia can work well together. And part of that is also because when I was training at Harvard, a lot of the PIs, including my own mentor, had companies. It was a very normal thing for a PI at Harvard and MIT to have a company. And so I saw that relationship between academia and industry, and it was very organic, and it seemed to work really well. So in my mind, I think it can be done. And so I do dance in both. I am an academic. I'm 100% academia, but there's another side of me. And I'm also a science mentor at Creative Destruction Lab for both the prime and for the cancer stream. So this is where biotech startup companies or startup companies pitch to investors their ideas or their innovations or their widget, and investors mentor them and invest. So I do that as well. But my forte is being an academic. But so there is opportunity to do both, and a lot of students do come to me because they see that I do have this executive MBA, and I do business stuff and mentoring and business strategy. They'll come to me and ask for guidance because not everyone wants to go into academia. I believe academia is an alternative career path for most people. And there's all these other things you can do with your science degree, particularly if you have a master's or a PhD. The way we think as scientists, we're very methodical, and at the same time, we're broad thinkers. We can be broad thinkers and be very focused. And we're able to, even with an honors student, their analytical skills are better than the average person. And so trying to convince young students who are not sure what they're going to do with their degrees that they could go into business or they could go into law, for example, is actually, these are viable pathways, alternatives to academia. Or, if you have a great idea, perhaps consider starting a biotech company. It's not easy, but it is doable. It can be done.
I think it's more common now as well to have it used to be more like if you're in academia, you're in academia, and where it's more, that barrier, I feel, is much more broken down these days.
And I think, I would hope that my colleagues and my more established colleagues are at least suggesting to their trainees that businesses or biotech is a viable option. And like I said, you could go in both directions. You could go, if you're a postdoc and you're not sure exactly what you want to do after 4 or 5 years, you could go into biotech. You could. If you don't want to start your own company, great, go into biotech. You may very well love the process, and you could grow and flourish. And if you don't like it, you can always turn around. And I think your point, Laura, is that you don't have to stick to one thing today like my predecessors did. And hopefully, my generation of scientists and academics are good role models for your generation to say, okay, I can move in this direction, I can move in that direction, and at some point, if I don't like one, I can move to the other. But having a higher science degree, like a master's or a PhD, I'm really serious when I say your brain is wired differently. You're able to be analytical, nimble, and quick. There's a tendency to see patterns before other people see patterns. And you'll notice this with your friends. You'll be out and you'll be doing things and you'll catch on to something a lot quicker than they may. And it's part because your eyes are trained to see things. Like we're looking at test tubes with DNA in them, but we can't see the DNA, but we trust the DNA is in there, right? We can see it. So we see things that maybe others may not see. So I think having the science degree gives us a better understanding of the world around us, I think. So I think it's okay if you decide to go into another direction. I would, applaud you for doing that. I have another student who went into law, and now they're a patent lawyer, but because they have a PhD, because they have this higher training, they're the best people to look at scientific inventions because they understand the technology. So you could do really well as a patent lawyer, right?
Yeah.
So I promote it. I promote going into different fields.
Yeah, sometimes as graduate students, we're not always introduced to all of these options. So it's a good idea to go see what's out there. And if not, you can always gravitate back to academia.
And I think universities have to have these science fairs, or even some of our programs could introduce sessions where you have people who have PhDs, who've done postdocs, they come in and they talk about their journey to become a lawyer, a patent lawyer, or a biotech person, starting a company or working in biotech, or even starting a business, or even starting a flower shop or whatever. I think having a scientific and an analytical mind will really help you.
Now, epigenetics is a field where there's many different definitions and interpretation. How do you personally define epigenetics?
So for me, epigenetics is the interface between humans and nature. And when I talk about nature, I talk about the nature around me, so the environment. So it could be the microenvironment, but most often it's the macro. So it's our environment, it's the air we breathe, the water, everything that we as humans interact with that can impact our biology.
Now, which aspect of epigenetics is involved in your work, or can you describe your research in your lab?
My research has multiple arms where cancer is the main overarching domain. We have projects that are unrelated to epigenetics, but one of our main projects related to epigenetics is we are trying to understand how there's a community here in Nova Scotia that has a very high incidence of cancers, and it happens to be a very old Black community in Nova Scotia, one of the original communities from the Underground Railway that arrived after slavery, and they arrived in Nova Scotia, and they've been in this one part of Nova Scotia for a very long time, and suddenly they're presenting with a high rates of cancer. And somewhere around 50, 60 years ago, a dump was built in the community, and the dump was used for military waste, and because they are on well water, they are also being exposed to this toxic whatever that's coming off the waste. And so my team is trying to understand the impact of this environmental waste, this dump, on this particular community. And so we're looking at epigenetic marks in individuals in the community to see if there is links, because we know some marks are related to different heavy metals, different toxins and carcinogens. And so we've got this project going to try and understand the impact of the environment on this community. And we've, part of this large grant for New Frontiers is with two other scientists at McMaster. And one of them is looking at the intersect of environmental racism in the community. And myself and the other molecular biologists were looking at the molecular biology elements of this project. But it really is a big epigenetic project, because the dump itself, where we know what chemicals, what carcinogens are in there. We also have mapped where their home is in proximity to the dump. And we've also got information of members of the community, the number of different cancers within a single family. And the surprising thing is it's not just one family on the block, it's every family has some sort of history of cancer, whether it's active, so there's someone with cancer alive, or family members who have recently passed away from cancers. So this is one of our large projects that we're really focusing in on the epigenetics in this community to see if the dump is the reason why. We're making the assumption that it's the toxic waste and the toxic groundwater that's the problem. And we're actually in the process of analyzing the data now. So hopefully within the next year, we'll have a report ready. And the other thing that might come out of this, because this is one of the first communities that this is being done in, we're creating this template on how to go into marginalized communities and do the sample collection and the consents. And we, building a relationship with the people in the community. And now that we've got this process in place, we hope that we'll be able to share it so that people who want to study other communities in Canada, particularly marginalized communities in the north of Canada or in other parts of Canada, to see if they can use our model to study some of the epigenetic events that are happening in different communities across Canada, other marginalized groups in Canada. So that is one of the more recent programs in my lab related to epigenetics. There's a second one that I started a very long time ago. Actually, it was at Harvard, where I was the first to discover a binding partner for the tumor suppressor kinase LKB1. And it so happens to be SMARCA4-BRG1, which is a sweet sniff chromatin remodeling protein, right? And it has taken us many, many years to try and understand this relationship between LKB1 and BRG1, because they're two proteins that are really difficult to work with. They're not easy proteins to work with. LKB1 is a little easier, but SMARCA4 is not. It's a very large protein. It's within a complex. It's very difficult to tease away its function from its the other SMARCA function. And thankfully, single-cell RNA-seq came on the scene and is helping us tease apart the role or the function of LKB1 and BRG1 binding together and what it might mean to histone modifications. And so this has been a very long project in my lab, and in part, it's taken time to tease everything out because the technology just wasn't there. And now the technology is. So, it's a lesson to my team. I always say, if you have a great idea, great, but the time might not be right because we don't have the technology to answer it. So don't throw the idea away, just set it aside and just hope that the technology comes around at some point. So that is the second very large project in my lab, is trying to understand this relationship, because we know that SMARCA4 is involved in a lot of chromatin and transcriptomic events. And so to figure out how LKB1 is involved in that, because they bind, we actually have mapped the domain. We've published this, so we know exactly where LKB1 binds to SMARCA4. We know that LKB1 enhances the chromatin remodeling of SMARCA4, and the kinase activity of LKB1 is required for that. So this has all been published. But what does it mean? What does it mean in the context of epigenetics, histomodifications? What does it all mean, right?
Yeah, knowing that LKB1 is a tumor suppressor, can you elaborate on the significance of LKB1 mutations on cancer progression? Is it something you're interested in to look into?
Yes. So LKB1 originally was discovered by Tommi Mäkelä and Lauri Aaltonen in Finland because they were on a mission to find the protein that was involved in a disease called Peutz-Jeghers syndrome. So they identify LKB1 as this protein, ends up being a kinase and a tumor suppressor, that's also really linked to Peutz-Jeghers syndrome. However, I come in right at the time they published that, and it was trying to understand, as a kinase and a tumor suppressor, what's its role. And so I was fortunate that they actually, when I wrote to them in 1997 to get the back then you had to write to people who clone things and ask them to share their clone with you. So they sent me the original LKB1 that they cloned from a patient, and then they sent me some of the mutants that were also found in the Peutz-Jeghers patient. So I have some of the original sets, and these are all these mutations all impact the catalytic activity of LKB1. So whenever we're working with LKB1, we're always working with the biologically relevant mutants that have come from either the Peutz-Jeghers patients or the ones that are found in breast cancer or in lung cancer. And then we have the classic KI mutation. So anyone who works with kinases know that if you mutate the required lysine to an isoleucine, you can kill the kinase activity. So we also work with the sort of the biochemistry modified kinase dead of LKB1. And so we're always, to understand the function of LKB1, you also have to look at killing its kinase activity, or what the role of the catalytic activity is in its tumor suppressor function. And in fact, they might be separate, because LKB1 as a protein also binds SMARCA4. It also binds the estrogen receptor. So that's, again, came out of my lab as well. Through mass spectrometry, we identified not only does the estrogen receptor bind to LKB1, we mapped the domain as well. So we know that LKB1 can regulate estrogen receptor signaling. So you have a kinase, but it also binds pretty important proteins like SMARCA4 and the estrogen receptor. So its catalytic activity has always been associated with its tumor suppressor function, but that might not be the case all the time. There might be who it's binding with and at what time, spatial temporal, who it's binding with at what point and where in the cell it's behaving, that might dictate its tumor suppressor function. And of course, we know that LKB1 phosphorylates AMPK on 3-172, and so that alone is the regulator of mTOR. So already you're thinking, this is it. This is the tumor suppressor function. Its ability to phosphorylate AMPK, and then AMPK reduces or blocks the activity of mTOR, and you shut it all down, right? But, I think there's still a lot to be learned with LKB1, and I think the next few years are going to, there's going to be some good discoveries with it, hopefully from my lab and from the other people around the world who work on LKB1 as well. So the mutants are important. To study a protein, you have to have its mutants, right?
Yeah, so maybe, turning a little bit away from the science now, how would you describe your mentoring style to the trainees in your lab?
So for me, it's always really important that I understand what their learning objectives are, what they want out of their career, knowing very well that could change every 6 months. Because how I mentor them also depends on what they want out of their degree, or what they want out of the experiment or that phase of their career. It's also very important for me I personally try and figure out if they need me to be hands off. They just need to know that I'm there and I'm available. Then I have to try and understand if there's someone who needs me to be on top of them, constantly asking them constantly, how's the experiments? What are you doing? Do you need help? Should we sit down? Let's have a conversation. So it's almost like a daily meetings and things. And then I have the people who just want to know I exist. And then I have some people who just want to have that morning meeting, that 3 minutes. This is what I'm planning. This is what I'm doing. These are my results. This is where I went wrong. This is what went well. And it's like a 5 minute in the morning meeting. And so for me, it's really important as the PI to not use the same mentoring strategy for everybody. Everybody's different. So you can imagine if you're someone who doesn't like to be micromanaged and you have a PI that micromanages you, it won't work. And vice versa. If you're someone who needs a macro manager, someone who's just in, like, I'm in the vicinity, and you're someone who wants micromanagement, then it won't work. So I think for me, I have to quickly figure out how this person best works or best functions. And that's how I mentor.
Yeah, I completely agree. To the ability to adapt, because not everyone has the same needs, and
and the needs change, right? As you get more proficient, as you move through life, as life challenges happen. So I think it's really my job to figure out how best to serve my people, right? To serve you, right? Because then when everything is balanced that way, I think this is where you get your best work.
Yeah, I think, as you know, when you're going through your career, even like from starting your PhD to like getting towards the end of the postdoc now, like the way you work changes, and the way you approach your work also changes, and it's different.
Do you think you become more efficient, or yes?
So yeah, but I think you also have a family. You have a family now, and like it's different priorities come in, and you work kind of differently. Yeah,
you work differently, but I think there's no loss of efficiency, because again, as scientists, doesn't matter what stage, honors, master's, PhD, postdoc, we're very nimble and quick. Our brains are very different. We're fast to move, we're fast to think, and you probably are doing as much experimental work as you were when you were younger, but you just have more on your plate now.
Yeah.
So you're thinking, how am I getting all the same? I'm still getting 2.5 days done in one day that I was doing as a PhD student, where I didn't have, let's say, different life responsibilities.
Yeah, definitely.
It's the confidence, it's how to move seamlessly. You know where to turn, where not to turn. When you're doing your training, you're figuring out where to turn and where not to turn, right? So I think as a mentor, it's really my job to figure out where you are in your needs as a scientist, right? The human stuff comes in, the person who has family and has other challenges is part of that too, right? And it's becoming even more part of it now. Thankfully, society is changing as well, so that becomes very important.
Now, before we let you go, do you have any words of wisdom for trainees who wants to stay in academia?
You have to love what you do. You have to absolutely love what you do, because it is hard. And we know the majority of the experiments fail. Like, we know that, right? This is a given. So you have to love what you do. And particularly when you're going through your training, your friends are going through all these other professional schools. So there's elements of FOMO in there, right? You're missing out on life. Your friends are moving forward. They're advancing in their careers professionally and financially. And here you are pipetting yet again on a Friday night, right? So it's okay to have that FOMO, but you got to love what you do, because ultimately what brought you to a lab was your curiosity. your love for discovery. You didn't know you had that until you actually got your first band or your first sequence. That's when you realized you loved it, right? So you always have to be brought back to that. Why did I start this in the first place? It's like what I said at the very start. The day I do not have that childlike approach to discovery and to research and that excitement that I have, it's pretty like, sometimes I have to dampen it a bit, but it will probably be the day that I walk away. So love what you do. And if it's science, great. If it's business, wonderful. If it's mathematics, fantastic. But love what you do.
Definitely. Well, thank you so much, Dr. Marignani, for joining us today and sharing your incredible insights into the world of cancer research.
Thank you. And thanks for the opportunity to share with the community some of my story. And I hope particularly the trainees walk away with maybe a different perspective on academia and research.
Thank you for listening to Season 3 of the This is Epigenetics podcast. If you like this podcast episode, you can check out our website for future episodes at thisisepigenetics.ca.